So welcome everybody to the November 4th meeting of the IMAG MSM Working Group on Multiscale Modelings and Viral Pandemics. We are 1 year old today. We've had more than 100 talks, and we appreciate very much people coming back. I hope that it's been a good year to be part of this for people. As usual, I need to remind you that this meeting is live streamed on YouTube and that the recordings are made available for public viewing later on. So please be aware of that. I actually have turned off the security for today because some people last week were having issues with it. We have two great speakers lined up for today. They both generously agreed to stay for conversation if there are additional questions at the end of the meeting. As always, you have myself, James Glazier, Reinhard Labenbacher, who is on study section today and is unfortunately not able to join us, Bruce Shapiro, who is here, and Jim Sluka, who is here, who are always eager to help out with any issues that you may have about organization, especially suggestions for future speakers or ways that the working group can work more effectively for you. We have our usual communication channels. Please help us spread the word about these meetings, especially in terms of the archived material and also the many white papers and discussion items that are being generated by community. We've created a Twitter account for the group on, please, MSM Viral. I hope that's memorable. If people could help us by retweeting the weekly announcements, that will help us build community. So if you need help with that, please talk to Jim Sluka about it. We definitely would appreciate it. We have great speakers, and we want to make sure that they have a good audience. Are there any announcements that people would like to make before we move on to the seminars today? I will repeat that I'm looking at the moment for 2 postdocs, possibly 3. Like many people, it's hard to hire good mathematical and computational biologists these days. It's a growing world. And so if anybody has a PhD student who's finishing or postdoc that's finishing that might like to work in this kind of multi-scale modeling, please send them my way. So again, it's our 1-year anniversary. Our first working group was on October 22nd. Our first mini seminar session was on November 5th. And so again, I exaggerated a little bit. We have actually had 86 seminars so far. So thank you all for sticking it out with us. Coming up next week, we have Lorenzo Veschini talking about image analysis and workflows for model generation from experimental images. We'll have Anna Bento, who's a professor here at IU Bloomington, talking about her epidemiological work. On November 18th, we have respiratory viruses and microbiome talk. No talk on the 25th for Thanksgiving. Then we'll have Claire Bryant speaking, and Reinhardt will be talking about the wrapping up of the crowdsourcing subgroup, which is getting a opinion piece submitted in the next few weeks. And we always have slots available for speakers. We'd very much appreciate your suggestions for people to speak. They don't have to be senior. We're quite happy if you have a good graduate student who wants to prepare for their thesis defense and make a presentation, or a postdoc who wants to present, feel free to suggest them as speakers as well. So please help us keep our meeting schedule full. Please try to remember to mute your microphones. Typically, we'll go through the talks together before the discussion, and then we'll have time for discussion. Both of our speakers have generously agreed to stay for the discussion session afterwards. And I will typically, if it looks like people are going to run over, I'll remind you 5 minutes before the official end of your speaking time, just so that you know where we are. So with that, we will start with our first talk on multiscale modeling of malaria parasites. As you know, I don't do elaborate introductions. We're very glad that Ulrich Schwarz has agreed to speak to us despite the late hour in Germany, taking advantage of the fact that this week there's a 1 hour less time difference than usual between here and Germany. And so I will turn it over to him.
Thanks a lot, James, and also Bruce and Reinhard for inviting me. So let me first fire up the presentation. So can you see my slides? Perfect. Actually, I made a last-second change here to agent-based modeling of malaria parasites because I'm not going to talk much about multiscale. Also, we are also interested in that. Today, I will give a relatively broad overview over the malaria field. And the only theory I'm going to mention is the agent-based part. So, but we also do multiscale. So let me start by somehow explaining how I got into this field. So the German Research Foundation, DFG, they have all kinds of different funding forms for large collaborative and interdisciplinary projects. And one of them is a so-called Collaborative Research Center, CRC. And we do have one here in Heidelberg, which is now already running into its 8th year. And it has been a very productive and exciting journey. And the subject is really very close to what you are interested in this working group. Namely, we look into quantitative analysis of pathogen replication and spread. And pathogens, in our case, is either viruses or parasites. So you can look up the webpage if you're interested in more details. So because 8 years is a long time, we already had several collaborative projects, some of them on malaria, some on other subjects. So we worked actually a lot on viruses. So I just flash a few published work on viruses. So we worked on HIV spread in complex 3D environments. This was a gigantic project stretching over many years. Then we recently published something on virus uptake. This is Rio virus from the ventral side, which is unusual because usually you look at the apical side. And there's a more theory-oriented work which just came out on the interferon system, which of course is triggered if you have a viral infection. So now let's come to malaria, which is the main subject of my work in this CRC. And I'm not sure if everybody is so much into it, so I briefly start with some basic facts on malaria. So it's really true to say that this is the most devastating disease we know. Of course, that's difficult in times of COVID, okay? But historically, it's certainly the one disease which has left its largest mark on our genome and on our society, okay? So if you start to look into it, you realize that a lot of what we are doing is shaped by malaria in some way or the other. So it still kills 400,000 humans per year, and I'm afraid the number might go up again, okay, because due to COVID, many processes have been disrupted. It's mainly children in Africa. If you're living in the developed world, there is drug which will cure you relatively easily. It's caused by a unicellular eukaryote, which is the genus of Plasmodium. And there are many different species, and 5 of them actually infect humans. So until recently, the book say 4, but now it's 5. Okay. So the most virulent one is falciparum. So Plasmodium falciparum is the one which affects you the most and often leads to death. But historically, Vivax was also important. Turns out that it's hard to prove, but probably Vivax was more common in Europe in the 17th and 18th, 19th century. So if you work on malaria, on the kind of work I'm going to talk about today, you often work with mice, and there the model system would be Plasmodium berghei. So malaria is transmitted by the bite of a female Anopheles mosquito, but there are many different species of these. And the same mosquito or other mosquitoes might also transmit other diseases, mainly viruses like yellow fever, dengue, Zika. And you see that these are emerging diseases, like Zika is more recent, okay? And there's a very special case, which is filariasis, which leads to what's called elephant disease, okay? The lymph nodes swell up. That's actually a worm which is transmitted. But today I talk about malaria, which is a unicellular organism. Now, the life cycle of malaria is pretty complicated. If you see it for the first time, it might confuse you, okay? So I hope you can see my mouse. Let me start on the left side. So basically a mosquito is taking a blood meal and it takes up one of the gametes from which is swimming in our blood, okay? Then it develops in a large cyst at the gut. It's really, so to speak, hundreds of these malaria parasites in the cyst, and the cyst opens up, okay? They basically swim to the salivary glands where they wait for the next blood meal. And then they are injected into the skin. And from the skin, they travel through blood vessels to the liver. And then it takes like 2 weeks. They come out of the liver again in very large numbers, okay? And they start to infect red blood cells. And there they go through a cycle. So for Plasmodium falciparum, it's a 2-day cycle, 48 hours, where they replicate from 1 to around 20. Then they rupture the red blood cell, infect new red blood cells, okay? And this leads to the periodic fevers we know from malaria. And they also develop some sexual forms, and these are essentially the ones taken up by the next blood meal. So, of course, there are many stages here, and for each stage, there are different questions one wants to ask, okay? And there is no unified model system for all of them. I mean, malaria parasites in different stages look very different, and you have to focus on one of these stages. So I will talk today about the skin stage, but we also work a lot on the blood stage, okay? So just a few remarks here. So we have 2 collaborators, one for the skin stage, this is Freddy Frischknecht, and one for the blood stage, which is Michael Lanzer. And I cannot help but mention that there's a little cartoon, the Malaria comic, which you can download from the internet, and both of them are actually characters in this cartoon. Anyway, so we worked a lot on the blood stage over the last years. And again, I just flashed some of the papers. So we started off by measuring the volume and area changes over the course of the 48 hours. Then we actually looked at the cytoadhesion, how they adhere to the vasculature to avoid clearance by the spleen. And very recently, we looked at the molecules underlying this. And this is where we really use multiscale modeling, where we go from molecules to cellular behavior. But today I would like to talk about the skin stage. And there the main actor enters, which is the so-called sporozoite. So as I said, in each stage, the malaria parasite looks different, okay? So this is the form you see if you look into the mosquito when it comes out of the cyst and goes to the salivary glands. And if you look into the skin stage, okay? And then in the liver, they already start to change into something different. So the sporozoite is a very long cell. It basically has a typical length of 12 micrometers, and it's always curved. So it's crescent, like a half moon, with a typical radius of curvature of 5 micrometers. And it looks pretty rigid, okay, because it has a relatively hard shell, but as you will see later, it's really flexible. But it has to be also rigid because it basically has to keep this very slender form. It has to compress the nucleus. It has a very clear structure. So at the front, you have this ring. So in the bottom images, you see the so-called, well, the ring which gives it the name apicomplexa. So all these from the same genus which have this ring are called apicomplexa. So toxoplasma would be another example. And then you have this basket of microtubules. And inside you have lined up a lot of containers with bad stuff, okay, which they basically use to actually inject into the host to actually get, I mean, to basically dissolve stuff and to open the way into the host, okay? Then in the middle you have the nucleus, or the sort of second third of the nucleus, and then some other stuff in the back, okay? So they move in the direction of the ring. And interestingly, they are sort of the fastest moving cells we are aware of. They are moving much faster than all the cells in our own body. And this probably serves the purpose of escaping the immune system. Okay, but how do they move? So this is an interesting form of movement. It's called gliding motility. And it's basically a flow of the surface from the front to the back. Okay, but at the same time, the surface sticks to the substrate with this trap-like adhesion receptors. And therefore, if the surface is stuck, the whole parasite moves forward because there's this constant flow from the front to the back. And then, of course, there has to be some kind of back transport for the system to actually have some kind of tank trading motion in the end. Now, if you look into the skin after a bite by the mosquito, you see that they are quickly moving out of the wound. And if you measure the velocity, you find this amazing number of 1 to 2 micrometers per second. That's much faster. It's 1 order of magnitude faster than every neutrophil in our body, which are the fastest cells in our body, okay? Which means that if they are really running like this for the neutrophil, there's no way to catch up with them, okay? That's probably the purpose of being so fast. That's also probably the reason why they are so slender and they can actually go like rockets through the collagen gels in the connective tissue. Now, if you actually superimpose the single images here, you get these trajectories and you see something very interesting, namely this kind of helical features of the trajectories, okay, something we are very much interested in. Then after a few minutes, they usually find a blood vessel. So here you see how they enter a blood vessel. So there has been a misconception for a long time that the mosquito hits blood vessels, which is not true, okay? The mosquito basically makes a large wound and injects the parasites into this, and then they have to escape the wound and get into the blood vessel. But there's no way the mosquitoes could hit exactly the blood vessels, okay? So once they're in the blood vessel, they quickly go with the flow and then they exit at the liver. So this is a quantification of how they actually go into the blood vessels. And here's another image which shows this helical features of the trajectories, which is a direct consequence of this chiral structure of the parasite. Now, they are helical in tissue, but if you put them on glass, they cannot escape into the third dimension, and therefore they run in circles. And one interesting observation is that they are always running counterclockwise. If you do it with an inverse microscope, as shown here. So if they run clockwise, they can't really do it well. So after half a turn, they basically flip over and go again counterclockwise. Okay, so there's some inherent chirality in these trajectories. The nice thing about this property is that they don't run away from your microscopic focus. Okay, so you can actually image them for a long time on without moving the microscope. And in order to mimic the situation in the tissue, which is pretty dense, full of collagen gel, collagen fibers, we basically came up with this idea to use these microfabricated chambers. So you see very nicely how they try to circle. Okay, and if the cylinders have the right diameter, around 10 micrometer, then they actually circle around the cylinders. And if the distances are different, they do different motion patterns. So these are some of the motion patterns you see. So if the cylinders are very dense, which is probably what happens in the tissue, very dense collagen gels, they more or less go straight because they really can't circle. But in 3D, this would be probably helices. So this is where we started to actually model some years ago. We use agent-based computer simulations where we put in basically the self-propulsion and the preferred curvature and some flexibility and some rules how they resolve conflicts in the labyrinth. And then we basically were able to get more or less the same motion pattern like we see in this pillar assays. Now, another thing I want to explain is that the motion looks pretty smooth, but if you zoom in, you see a lot of stick slip. Okay, so they get stuck from time to time, and then they use force to rip themselves off. And as you can see on the right-hand side, they deform a lot. Okay, so this is the first time I really show you that they are really flexible. Okay, they are stiff locally, but as a large object, they are pretty flexible, and this is very important for them. So we measure this with high time resolution, and we see this typical stick slip motion. And to understand better the forces, we use traction force microscopy. This is something we do a lot in my group, okay? And you see that if they get stuck, they actually start to pull on the substrate until they rip themselves off, and then they have a very fast relaxation movement forward, and then they go back to the steady state. Okay, so they are really geared towards fast motion. If something holds them, they basically rip it off. Now, one thing we discovered more recently is that if you actually look into the mosquito stages, there are two stages where they are forming large collectives. Okay, this is the cysts at the gut, and then later they accumulate in the salivary gland. So what we managed to do is actually we gently dissected the salivary glands, and then we basically get these collectives free on our substrates. Okay, now you see that the microscope is the other way around, so they now rotate to the other direction, but it doesn't matter. And this way we actually could look at the mechanics of these guys in this context of collective motion, which is a pretty new angle on the system, okay? Because usually people don't look so much into the mosquito stages, but by doing this, you get these collectives, and then you can learn more about how the sporozoite moves in a complex environment, which is happening in the skin stage, okay? So we then used quantitative image processing, again, in the framework of this CRC. We always have collaboration partners who help us. Okay, so there's a group, Karl Rohr, who works in the image processing. We segment out the shape of the parasite from the GFP, which is expressed, and the nucleus we can segment out from a Höchst signal, and then you can very nicely reconstruct the trajectories of these guys. And because they run in circles, you can then actually show the midpoint of the circle, which you see in the middle, okay? And you see that they basically cluster, which means it's a vortex where they basically all run around the same center. So then we actually sit on the vortex middle, go radially out, and record the velocities, okay? So you see that the angular speed goes down as expected, okay? The linear speed is to a first approximation constant. But if you take a second look, you actually see that there's a difference between small and large vortices, okay? Turns out that if you go from a small vortex, which is the blue line, to a large one, which is the green line, there's a change from more or less plateau, or maybe slightly increasing, to a biphasic relationship, okay? So something happens if there are many of these guys together, they sort of sort according to some criterion, and we think it's basically speed, okay? So in order to understand this better, we turn to agent-based modeling, and now we have a more mechanical model. So basically we have a chain of beads which define a certain bending stiffness. Each bead is self-propelled in the direction of the tangent. This gives a motility force, okay? And we have some rules for collisions. Like we look at the movies, we see there's alignment and there's crossover. So we put this into our agent-based modeling. And most importantly, we realized that we really have to put in a distribution of curvatures and motility forces or speeds, otherwise it doesn't work, okay? It's really important to understand that each sporozoite is a little bit different, okay? And if you take a monodisperse solution, it would not work, okay? So you need this distribution, which we have measured more or less experimentally, but in the simulation, we take 2 Gaussians, okay? So we now run these computer simulations, and you see we get very nicely these rotating vortices. It's periodic boundary conditions, and then we can vary model parameters. So here we change the repulsion. This is the rule if they hit each other, okay? And we change flexibility. And if you have a lot of repulsion, a lot of flexibility, this is the top right, you get very large vortices, okay? And otherwise you get smaller ones. So we now can actually measure do the same analysis we did before. We can measure the angular speed and the linear speed, and indeed we get similar curves like before, okay? And we can go back to the model and change things and figure out what really was important. And as I already said, it's really important to have this distribution of curvatures and speeds. So they actually sort themselves out, okay? And the most important element is definitely flexibility, okay? Without flexibility, they could not form this vortices as we see them. So this is a direct proof that they have evolved a high degree of flexibility, which probably is very important in these mechanically challenging environments. to start with in the wound and in the skin. So with this, I come to the end. So I hope I was able to give you some flavor of what we are doing here in the skin stage. Malaria parasites go through many different stages, and each stage is different. Okay, so I can say that because we also work on the blood, and it's a completely different story there. Okay, but here in the skin stage, you see that there is this very impressive achievement of the parasite to run extremely fast through the tissue. Okay, and it manages to do so by being very slender. Okay, by being mechanically very flexible, by having this amazing surface flows. Okay, and having the right adhesins on the surface. So I think basically they have evolved to outrun the immune cells, okay, and to find blood vessels. And we think it's not a coincidence that the typical diameter of blood vessels and the typical curvature diameter of sporozoites is pretty similar, okay? So we think that they actually use this to curve around blood vessels until they found a good spot to enter. And I also want to mention also the collective phenomena is a little bit artificial in the framework of the skin. It's really important in the mosquito stages because there you have two stages, the oocytes and the salivary glands, where they do have these collectives, okay? Also in the salivary glands, they can't move because they are stuck there in small cavities. So I mentioned our collaborators along the way already. And in the end, I would like to acknowledge the different funding agencies. So this work I was talking about is the CSC 1129. And as I mentioned earlier to James, we now have in Germany a new initiative, a so-called priority program in physics of parasitism. And I'm really excited to be part of this because we will look at many interesting systems in comparison, like toxoplasma versus malaria and so on. So thanks a lot for listening, and I'm happy to take some questions.
Okay, thank you very much. It's wonderful biophysics of fluids, the way microorganisms move in fluids collectively is always a fascinating problem. So our usual policy is that we go on to the next slide,
okay.
And then do the questions at once.
Okay, fine.
Okay. So our next talk will be from Stanka at Virginia Tech, who's going to be talking about the role of testing in COVID-19 control. And I will turn it over to you.
Thank you. Can you see my screen? Okay. So I want to thank James, Reinhard, and Bruce for inviting me. And what I'm going to talk to you today is work my group has been doing in the last year and a half on COVID-19. And in particular, I usually work on it within host models. This is a multi-scale model work, and it has to do with testing. So the idea behind the work started in the mid-2020, and it came with me participating in a lot of our workforce here at Virginia Tech, in which we were trying to see, can we quantify the amount, the type, and the frequency of tests that are needed in one community. So this was before vaccines were available. And by community, for my case, we're talking the New River Valley around Virginia Tech, but what I'm going to show you in here is the testing in Virginia. So every bar that you're seeing in here is how many PCR tests were administered every week since March 2020 up until last week. And then everything that you see in light blue is how many of them came out positive. But just as a zoom in, if you're looking on the right-hand side, this is the percentage of tests that are positive, and the yellow curve gives you a 7-day movement. So the idea is that, this is how many tests we're doing, this is how many of them come positive, but these are all PCR tests, which are expensive. They are amazing in the sense that they are very sensitive to the virus, but they are more expensive. They require a lot of training for medical personnel. So when it comes to Virginia Tech, or even when it comes to smaller communities, you have alternatives. And what we were trying to see is, can we quantify, can we use mathematics to quantify these alternatives? So let's talk a little bit about these alternatives. So if you are thinking about just diagnostic, that means someone comes in with symptoms, they probably get it, but you're going to use PCR or an antigen test to confirm that they are positive. But when it comes to surveillance and screening, you have lots of options which Virginia Tech is employing right now. Like we measure wastewater from our dorms, we do pooling. But in other words, you can have other tests with less sensitivity, but that can give you good results, especially at the peak of someone infectiousness. So the punchline of what I'm trying to say here is that one type of test doesn't fit all communities, and this has been very nicely presented in a science opinion paper by Michael Mina. And the other point that I'm trying to make, and this is some work we're doing at Virginia Tech, is that when you're measuring virus, when you're using PCR to measure virus, what you're measuring is RNA. And if you're looking at the differences between RNA and infectious virus, they are huge. So this is a study that's been done not in humans, but in hamsters by Nisha Dugal, a virologist at Virginia Tech. And what we're trying to measure in here is the aerosol virus. And we're doing this with Lindsay Moore, which is an aerosol engineer. And what they are doing is they are putting hamsters in a closed room, and they are trying to see how many variants they exhale. But for the purpose of this talk, the only thing that I'm going to take out of this study is the difference between the RNA and the infectious virus. And you can see that there's a lot less infectious virus. Those are the red dots and the red curve compared to viral RNA. So in other words, you can overtest at some point, right? You can test somebody that is no longer infectious, like this example over here. And just to give you an idea of what big the difference is, if you're looking at RNA over infectious virus, this is log 10. So you can go all the way to 3 orders of magnitude difference between them. Okay, so our first study that looked at the role of testing was published in Viruses, and it just looked at the trade-off between test sensitivity and test frequency in COVID-19. And for the remainder of the test, frequency for me is going to be cost. In other words, if I have $50, how many PCR tests versus how many other types of tests can I do with the same amount of money? Okay, so in order to figure out how the tests behave and how sensitive they are to the body, to the virus inside of a human, I need to actually connect the test sensitivity with the amount of virus inside of an infected individual. And there are very few data sets, human data sets out there. One of them is the one that I'm going to present in here that comes from an outbreak in Germany. All of the patients in this study were in their 30s, healthy adults. The other one is the NBA bubble, again, very healthy adults. We know that athletes that would do really well with health tests and things like that. But the idea is that if you're looking at this outbreak and you're looking at the amount of virus in the upper respiratory tract and lower respiratory tract, you can put together a within-host model and you can quantify the amount of virus there is in a patient, and then you can put testing on top of it and determine at which time points during the infection someone is transmitting. Okay, and I'm not going to show you the data, but I'm going to show you what kind of viral dynamic I'm getting out of there. So in red is the virus in the upper respiratory tract, and that is the one that I'm going to be using because that's what the PCR measures. And in blue is the lower respiratory tract. And with that, I can look at what testing does when I'm assuming that I have this kind of viral load. And again, at this moment, I'm assuming everybody has this viral load. And we can talk about, later on about how sensitive this is to different types of infected individuals. So here in the first two panels, I'm talking about a PCR. So if I have a PCR that measures the virus, this is the period of time in pink in which an individual is infectious. But this is everything above the red line is something that the PCR catches. So in other words, I'm catching someone when it's he's not infectious at the beginning, or he's not infectious at the end. But the idea is that if I'm using this for diagnostics, so this is where I'm going to test this individual, and I have, and I have a delay in my return of 5 days, which at the beginning of the epidemic was a very common outcome. I will tell this patient at this moment that they are infectious, and I'm only going to save them from transmitting for a very short period of time. But even if I'm using this for surveillance and I'm testing them before they are actually infectious, and I am allowing the long delay for the return, I'm still only saving a little bit of time when this person is transmitting. Now, if I'm doing an antigen test, here is the sensitivity of my antigen test, right? So I'm missing a lot of time in which this person is infected. However, if I'm using the antigen test and I'm doing it frequently enough, then I can catch someone right after they became infectious. An antigen test returns the result in 15 minutes. So I am saving a lot of infectiousness period. Okay, so that, as a cartoon, I hope everybody that makes sense for everybody, but can we actually quantify it? Can we put a mathematical model on top of it and see when does it make sense to use antigen tests versus non-antigen tests? So what we did is we did a continuous model. This is a PDE with delays because I have to account for the delay in test return, in which I have a susceptible population, and then I have ages of infection. So I have infected individuals that are asymptomatic at different ages of infection, infected individuals that are symptomatic at different age of the infection, and then I'm assuming that I'm isolating them the moment they get a positive test. And I'm starting to test, and if I'm testing with an antigen test, right, I might miss it, but eventually I catch it and I isolate them right away. And if I'm testing with a PCR, I might get it right away. I might get it right here, but then I'm going to have a delay in test return. And this is when I'm going to isolate them. And I'm going to assume that I know how many tests I have. And this is the capacity rate. And for me, is a percentage of the population. I know that I can test, let's say 10% of my population each day or something like that. And I can transform that into a testing rate. And with those, we wanted to see what kind of results we're getting. And we did really high testing. We can do lower testing, but at the time of the study, we were interested in what happens on college campuses, and the level of daily testing on college campuses was really high. So we assumed 10% testing each day. And I'm going to start with this. So in pink, you see how many cases I have, and in green, you're going to see how many of them I'm catching, right? So if I'm doing a PCR test, I am getting almost everybody. And I'm removing them from the infectious population with a little bit of delay. If I have antigen tests, then I'm catching a lot less. But if I'm looking at the daily incidence, I'm flattening the curve. So if my goal is to flatten the curve, then antigen tests actually do a lot better. And this is one-to-one, right? For each PCR, I'm doing one antigen. But the point is that I don't want to do one-to-one. I want to do more antigen because they are cheaper. How much cheaper? Well, before I jump there, let's actually put the numbers. So here's my antigen test. So this is around 46% of my population being infected. When am I getting something similar with tests of decreased sensitivity? Well, if I have a PCR, I'm getting exactly the same ballpark. If I have a return delay of 4 to 5 days, in other words, use a PCR if your return delay is smaller. Now, if you have a saliva test, you do that with 2 to 3.5 test delay. So use this for any return delays that are smaller. And then we can do this with a test that's sensitive to the point that you're only catching things about 10 to the 4. And I give you, I don't have a test that is available right now for that. But if there was, you would get an information from there. But the idea is I don't want to do one test to one test. I want to do many antigen tests compared to an RT-PCR because they are cheaper, right? How much cheaper? So we came up with a graph. So this is the graph that tells me if I have a fixed budget, how many PCRs can I do for the same antigen test? And we were doing Abbott Now at the time. So the PCR test was $50. Actually, Virginia Tech pays more than that. The Abbott test was $5 per test. So I can do 10 Abbott tests for one PCR test. And if I'm doing that and I'm plotting again how many infected cases I have for the antigen, that's like 1.2% of the population, and I'm comparing it with an PCR with 5 days delay, I'm doing way, way better. But even if I'm comparing it with where we are right now, almost all cities do a PCR return of 1 day or less than 1 day, I'm still removing 10 times more people with antigen test for the same amount of money. And then you can look at things on where does this break? Where does this analysis break? So in blue, you have the PCR. In red, you have the antigen test. And now you can say, what if I'm trying to save money and I'm doing half of the antigen test or a third of the antigen test? I'm still doing better than the PCR. The only time I'm doing worse is if I'm doing things that are usually, this will be, once a week antigen test versus testing everybody once a week versus testing everybody once a day. Okay. When we were doing all these tests, there were no vaccines. And of course, vaccines are available. People are getting vaccinated, which is fantastic. What does the testing do now that you have the vaccines? So these are the vaccines in the United States. This is the efficacy at the time of the vaccine approval, EUA approval. And this is the amount of vaccines that were given at the time. So we have a lot of Pfizer and Moderna. We had some Johnson & Johnson before it got discontinued. And but I want to connect this with testing. So I want to know what the vaccines do in terms of the level of the virus inside of the body. So this is a test, this is a study that came out of Israel that looked at CT values. So think about CT as being inversely proportional with the viral load. So if you have a high CT value, that means you have a lower viral load compared to a high CT value. And what this study showed was that if you're getting and they only looked at Pfizer, almost everybody got the Pfizer vaccine in Israel. So if you're looking at the vaccine for the first 11 days of the vaccination, you get levels that are very similar to unvaccinated people. So I'm talking about breakthrough cases here, right? So you get the vaccine and then you got the disease. If you did get a breakthrough case and you were in the first 11 days after the vaccine, your viral load was the same. But if you did get a vaccine past 11 days, then the viral load was decreased by 4-fold. So we want to put that into our model. So this was my original model. This is my viral load. Now I'm vaccinating people. I'm still considering the age of vaccination. For the first 11 days, they look like unvaccinated people. For the days after 11, they look like vaccinated people with a viral load that's green. So that is 1/4 the original viral load. And now I can consider breakthrough infections, and I can again look at testing. I'm not going to consider antigen testing anymore. I'm going to consider everybody gets a PCR test at this moment. Okay, and now we want to account for. So I'm saying, right, the vaccines decreases the viral load. Now, what do changes, variants of concern do to the vaccines, right? Because I need to have a feedback between the two. And I was looking at the alpha variant, so that's the variant that came out of UK, and the delta variant that came out of India. And what we know in terms of the delta variant is that it has an increased infectivity. It's reported everywhere between 1.5 and 2% were used, an infectivity rate that was increased by 1.6 completely. I don't have a reason for that, and we did sensitivity on that. And then in terms of the vaccine protection, here's the alpha vaccine, which is the protection at the beginning of the EUA. And here is the Pfizer vaccine for the Delta that came from a study in the UK. So basically the first dose was decreased to 33% and the second dose was decreased to 88%. And now I can look at several things. So the first thing that I'm going to do is no testing whatsoever. And I'm going to look at how many breakthrough cases I have with an alpha variant and how many cases I have with the delta variant if I have an outbreak that's huge. So I'm assuming 5% of my population is infected. And what you're seeing in here, these are percentages. So I have 0.72% for vaccination rate that is, this is the background vaccination in the population. So I'm assuming a population that has 10% of the vaccine, 10% of the population being vaccinated at the time of the outbreak versus 80% of the population being vaccinated at the time of the outbreak and everything in between. And this is the additional vaccination per day. So of course, I'm doing better with the breakthrough cases if I am starting with a highly vaccinated population and I'm still giving extra vaccines at the time, compared with a low vaccinated population. This is with the alpha, and these numbers become a lot bigger when you have the Delta variant. But you can see that the breakthrough cases are still relatively low if I have a highly vaccinated population. But I'm interested in testing. So what does testing do? And not only I'm interested in testing, but I'm interested in determining whether or not testing everybody or testing non-vaccinated, which is the way we're doing it. For example, at Virginia Tech, we're only testing the non-vaccinated students. We have a vaccine mandate. So which one gives me better result? And it turns out that if I do this in the alpha, in an environment in which alpha is dominant, the differences are not very big in terms of numbers, but I'm seeing, so again, this is the background vaccination level, and then this is testing capacity. So if you're going from the left to the right, you are decreasing, this is differences between background infection and what you're getting in the presence of tests, right? So if I have a 10% vaccinated population, 63% of the population is infected. And if I'm testing 5% of them every day, then I am going to reduce that by 0.28. But I'm not doing, if I'm going from, the same amount of testing, but I'm doing this in highly vaccinated population, I'm doing worse and worse with testing, to the point that for the 80% vaccinated population, I am reducing the outbreak by very little. Now, of course, this is a very little outbreak as well. But when I'm looking in the Delta case, then things are a little bit different. In particular, if I'm going from left to right, I'm increasing the number of infections that I'm preempting. But if I'm going from down to up, I don't have a decreasing number. On the contrary, I have a peak somewhere in the middle. In other words, I'm doing a lot better with testing when I have some intermediate vaccinated population compared to high and low vaccinated population. So what does that mean? So instead of looking at Delta and Alpha, let's look at variants of concerns of higher and higher infectivity. So this is the infectivity rate. Alpha was 0.25, so somewhere in here, and Delta was 0.4. What if I'm getting a new variant that becomes more and more infectious? What's going on? So here I'm having the percentage vaccinated population, and here I have reduction in cases for all of these. When I'm assuming that I'm testing everybody, 10% of my population every day. And what you're seeing is that even though I'm doing a lot worse, the higher the infectivity rate is, I am doing better when I am using testing in highly vaccinated population than in the absence of highly vaccinated population. And as before, I'm doing better if I'm testing non-vaccinated compared to testing vaccinated. So in this case, even though only 20% of my population is not vaccinated, I'm still doing better in reducing cases, at least percentage-wise, if I'm testing for in the highly vaccinated population. The other thing that we did was, I'm assuming, in this case, I'm assuming that the infectiousness doesn't affect the vaccine. So I have a vaccine that's really good. But what if infectiousness leads to a decrease in vaccine efficacy? I'm still, so what I'm having in here, I'm assuming that I have 50% of population being vaccinated, testing only non-vaccinated or testing everybody. And then I'm looking at the efficacy of the second dose that goes down from 90% all the way to 40%. I am doing worse for a highly infectious virus compared to a less infectious virus, but I'm still doing better if I'm testing in highly vaccinated population compared to lower infected population. Okay, so I'm going to conclude here. I'm going to say that testing is still a powerful tool for outbreak prevention, but the value of testing diminishes as vaccination becomes more and more widespread, with higher effectivity of emerging variants being the highest factor in determining where the peak of test efficacy is. Vaccination provides better long-term control. You saw that the testing reduces over time if I have a low vaccinated population. And then when you're considering vaccines versus testing, vaccines are more important, but there is a little bit of trade-off between them. And putting together an optimal control model might be important. And I'm just going to conclude with this. I was asked to talk about big picture things that I'm thinking of. So some caveats in our model that I'm considering, and I would be very interesting to hear people's opinion about it. So first of all, when we're looking at the viral loads inside of an infected patient, with the very few exceptions that I showed you, what people look when they compare vaccinated with non-vaccinated or alpha versus delta is cross-sectional studies. Meaning I'm taking a CT from a patient, I'm saying this patient is at day 5 in the infection, and then I'm taking another patient at 7 point in the infection, and then I'm assuming I have a population kind of viral load, but it comes from a different individual every single time point. That's not even the thing that worries me the most. The thing that worries me the most is that it comes at different portions in the epidemic, in the pandemic, right? So people behave a lot more different in 2020 than they behave now, than they behaved this summer when it seemed like we had the pandemic under control. So can we use these statistics when we compare levels between vaccinated and non-vaccinated and alpha versus delta? The other thing is we're asking vaccines to do this fantastic thing, which is reduce transmission. But really the vaccines for other diseases have been developed in order to prevent disease. And I'm just showing you a difference between cases in non-vaccinated in black versus vaccinated in blue. And if you're looking at breakthrough infections, yes, the blue goes up. But if you're looking at hospitalization and death, they stay constant even when a new variant comes about. So how do we incorporate that into the model to think about this from the point of view of having a disease rather than stopping an infection? And with that, I want to thank my collaborators. I did the multi-scale models with Jonathan Ford, and I'm doing the in-house models that give me the infections versus non-infections with my graduate student, Nora Breen, and with data from Nisha Dugal. And I want to thank funding that comes from NSF and from Center of Emerging Zoonotic and Anthropod-borne Pathogens at Virginia Tech. And I'm happy to take questions.
Okay, thank you very much to both of our speakers. About as diverse a pair of talks as we could have had, both fascinating. And I will open the floor to questions.
Can I ask the first question?
Please.
Can you explain again what kind of measures you have for viral load? So, I mean, I had the impression you talk about 2 different measures. Of course, you always have the PCR or the rapid antigen tests, which basically look for RNA content. Is there any other measure you are considering?
So I was considering 2 things. So first of all, I was considering the RNA measure. And in the patients that came out of Germany, you're probably more familiar with this study than I am. They did RNA per swab, right? That's what they have. And then when I was putting on the cartoons, I was putting the pink bars that look at where does this patient transmit, right? Because infectiousness is important. Those bars come from studies, culture studies that look at how many RNA do you need to have in the body in order to get a virus that is transmissible, right? So it's two different studies. I don't have the actual infectious viral load from any of the human patients.
Okay. And the second short question I have is, I mean, you mentioned that there's a large range of the efficiency of the rapid antigen tests. You said it goes from 100 to 100,000 viral RNAs per swab. So, I mean, where does this large range come from? Is it different manufacturers, or is it the way you actually perform the swab?
No, so basically, I'm assuming that the antigen tests detect. So if I'm looking at the numbers, they detect around 10 to the 5 RNA level.
Yeah.
And everything else was, I was trying to see what if I have tests in between with more sensitivity. So that was purely theoretical. The only things that are real world are the extremes, right? PCR detects around 100, and antigen test detect around 10 to 5, and everything in between was, if you had a test like that, how much do you, how much testing do you need to do in order to get the same kind of ballpark numbers?
Yeah, thanks.
Sure.
More questions for either speaker? As always, I have a lot, but I like to save mine until everyone else has asked theirs. Well, maybe I'll start off one for Ulrich. It's something you didn't talk about, which is I found the biophysical discussion fascinating. What is known about how the malaria parasite moves from the tissue to the bloodstream? How does it actually enter the bloodstream? And then maybe as a second question, how does it target the liver and find its segregate to the liver?
Okay, so there is a clear-cut question. There's a clear-cut answer to the second question. Okay, so for the liver, it's more or less known how they do it. There is a certain cell type which I think is called Langerhans cell, and it basically connects the liver to the blood. And they somehow manage to get into these, and these are really like bridges. They just run through them into the liver. So that's what the textbooks say. And for the first question, there is no good answer. We don't know. Okay, so it's probably likely that they have to. I mean, it looks from the microscopy as if they had to search for the right places. Okay, there must be some weak spot, but I'm not sure. We don't know what this weak spot is. Okay, I think the one thing which I learned is that there's no real chemotaxis there, okay? And my explanation would be that the mosquito creates a really bloody situation with a lot of blood flow and debris and so on, and it just doesn't make sense that there are clear diffusion gradients there, okay? So I think this is the reason why the sporozoite has to rely a lot on mechanical cues, because chemically it's a very disturbed environment. But if it comes to the blood vessels, there might be a better situation. And one hypothesis would be that maybe it senses oxygen, okay? But as far as I know, this is not known.
Are there specific adhesion receptors on the parasite surface that recognize the target adhesion molecules either in vasculature or in the liver, in the Langerhans cells?
Yeah, surely Langerhans cells, yes, but the other way around, I'm not sure, because if you come from the inside of the blood vessel, it's easy, okay? It's like a white blood cell which wants to extravasate. There are many known adhesion receptors starting with the selectins, okay? But in the other situation, you come from the outside to the blood vessel, which is, nobody's doing that, okay? That's an unusual situation, and I'm not sure which cues you would exactly use in this case, so I don't know.
If I can follow up on that. So why is it that the parasite moves in circles? And also it seems like it's cyclical, right? Is there any evolutionary reason for that? And does that make them more fit or less fit? And can we take advantage of it in any way?
Yes, exactly. So it fascinates us as it does you. Okay, so it only runs in a cyclic manner if you force it on a glass slide, which is artificial. Okay, so in real life it runs in helices, and there's of course the mathematical biology literature on search strategies is large. Okay, and there are ideas that sort of, I mean, there are motion patterns like tumble and run and so on. Okay, and I think the circular pattern is not a bad idea. Okay, it could be a good strategy if you look for islands like infotaxis. I think Boris Schreiman has a paper on infotaxis. Okay, so it might be a good strategy, like all random walks actually, to combine local searches with more global propagation. Okay, but still, I don't know what exactly is optimized there. But it must be very profound because the parasite has evolved this. Okay, there's a clear machinery. It's basically the ring plus the microtubules plus the adhesion system. It creates this helical motion pattern. So there must be a good reason for it. So we like to think that it has to do with the blood vessels. Okay, circling around the blood vessels in the end. But maybe it's also an advantage going towards the blood vessels and sort of exploring space in a intermittent manner between local and global. Okay, but it's all speculation. Nobody knows.
Fascinating.
I mean, just adding, you see, the sporozoite is the form of the malaria parasite, which you have in the oocysts, in the mosquito, then in the salivary glands, and then in the skin. Okay, so it's not only optimizing something in the skin only. Okay, it could be that it's an optimum over many stages. Okay, so in the salivary glands, you have small cavities where they have to smuggle in high densities. Okay, maybe the crescent shape is good for packing reasons in the salivary glands. Okay, so maybe we are doing the wrong thing if we look for the optimal in the skin, and the real optimum is in the salivary glands. So it's just very difficult to answer this. But I think it's not a coincidence that the diameter of the blood vessel and the curvature radius are very similar. Okay, looks not like a coincidence to me.
I guess, I mean, in some ways, if you're swimming, it's hard not to move in a spiral. You have to work to not move in a spiral, because if you have any asymmetry in the propulsion, it's going to tend to create a spiral movement. I'm thinking of things like listeria, whereas here you have very clearly a curvature in the organism that is tuning that spiral, which is
but it's not swimming. The problem is not swimming. If they are in fluid, they only move passively in the hemolymph, in the mosquito, and in the bloodstream. So it's really an adhesion-based mechanism. And yeah, but I think moving through a porous medium like the connective tissue, probably a spiral is a good idea because it gives you a lot of traction. Okay, and if you have a spiral, you can store some elastic energy, basically, which you couldn't do in a straight rod. I think this is one of the things I think is happening actually.
Well, you made the comment that you want to outrun your neutrophils.
Yes.
But presumably that would be advantageous for many pathogens besides malaria. And so it's interesting that must not be the solution that they found can't be an easy one to find, or you would see it more frequently.
No, I completely agree with you. It's the malaria is really the fastest one, and you can ask why are the others not as fast? Yeah. Good question.
Other questions? Usually we have a lively audience. So.
Very naive. Is this can be explained by similarity to swarming? Like
you mean the vortices I talked about?
Like what we see in fish school?
Yeah. Well, you see the vortices I showed you, the rotating vortices, they are a little bit artificial in the sense that we extract them from salivary glands, and in salivary glands, the sporozoites do not move, okay? Because in salivary glands, it's too small a space. They are basically waiting to be ejected. So there the packing is probably more important, okay? And then later, if they go into the skin, they are alone, okay? So the point is that the mosquito, let's say the other way around, the parasite, make sure that not too many of them go into the skin, okay? So typically 10, but not more, okay? And from these 10, I mean, at most one will make it to the liver, okay? Many of those will run into lymph and they die, okay? They're running out of ATP and they just don't make it. So it's really a very scarce event that they make it. In the skin, they are basically alone and not in collective. So the collectives I showed you are a little bit artificial in the sense that they don't occur in nature in exactly this manner, okay? So we shouldn't attach some function to it. We use the collectives to learn something about the parasite, but we know that in the skin, it's a lonely parasite, okay?
But the helixes that you see in real life, and you have very good explanations to the differences, I can view these helixes as stretched spiral.
Yeah.
Or stretched circular motion that at some point I pick it up and stretch it to make it three-dimensional.
Yes, right. That's true. And I mean, it's what we learn from the 2D assays is that they only can run counterclockwise, okay? So if they run in the wrong direction, they basically pick up some kind of strain or something which ruptures them from the substrate, okay? And this can be different in 3D. Maybe this is relaxed there. So this is something we want to study in the future. I mean, if they're really, if there's the same strong requirement on chirality in 3D than in 2D, we don't know, okay? So maybe they use this actually as an invasion strategy. I mean, to actually, I think maybe there is a lot about mechanics here that they want to push themselves into some complicated mechanical situation. Maybe they even store some elastic energy in these spirals.
Thanks.
Go ahead.
Have you noticed, you've mentioned that they just go in spirals, but you've mentioned earlier about tumbling. Have you noticed any where they're just spontaneously flip and go the other way? Or you've mentioned that they can flip themselves over, so they can go counterclockwise. Is that just a random phenomenon where they flip over, or do you think they purposely flip themselves over so they can go counterclockwise?
Yeah, I think they purposely do it to resolve complicated situations. So if they can't get ahead with the usual gliding motility, there seems to be a way that they basically use their rear as a foot, okay, and from there to explore new directions, okay, really like tumble and run if you wish, okay, and then to find the next best situation to continue.
Okay, and then my follow-up question to that was, I might have missed it, but did you guys, you have the slide with the little, I forget what they are, but basically the two-dimensional slide where they just go in circles.
Yeah.
Did you guys do any chemotaxis or like put blood or something to see if they move one way versus the other towards some stimulus?
No, we didn't.
Okay.
No, we didn't. But there's no, as far as I know, there's no chemoattractant or chemorepellent known. Okay. So to my knowledge, it's not known. And by the way, there are other important systems like human sperm where there's also very clear directional motion, but no chemoattractant, okay? So, I mean, in sperm, it's, I think, more temperature taxes, for example, okay? So it need not always be chemicals, okay? And I think for the sporozoites, mechanics must be very important because it's a mechanically challenging situation.
Thank you.
Other question. I was going to say, you mentioned something, Ulrich, that I think was quite significant, which you already started answering my next question, which was about optimization. You said that the parasites, of course, have a limited ATP, supply. And I was going to ask you about the energy budget. In other words, how much of the parasite's energy budget is devoted to being able to move so fast? And you could certainly do a nice agent-based trade-off model, or even a non-agent-based one like the first passage time of like Stanka's model, where you said, okay, if I'm being pursued by neutrophils, and the faster I go, the less likely I am to be eaten by neutrophils, but also the faster I go, the more likely I am to run out of energy and die before I reach the liver. You could do an interesting trade-off model to ask the question about optimization for the speed of migration.
It's a very good question, yeah. But let me mention one thing. So, because I mean, I have a lot of expertise on ectomyosin, okay? And this is an ectomyosin-based system, but it's completely different from what we know, okay, in humans, because the actin is very short. It doesn't form filaments or networks. It only has sort of very short strands of actin, which form something like a fluid, okay? And the myosin is like a myosin 1 in humans. But it's still actually a matter of debate where exactly the myosin is located. I mean, it's amazing that we don't know exactly if the myosin is attached on one of the membranes. Okay, there's a membrane system below the plasma membrane, or if it's some other, it's just very difficult to find out, okay? So, I mean, the energy budget on the molecular level is not clear, because we don't really know if there's a lot of actin polymerization, which might use actin, okay? We don't really know what the motor is doing in detail, okay? So on a molecular level, we would not be in a good shape to write down an energy balance, okay? But of course, you're right, we could do it on the level of the cell. We could talk about friction and efficiency, like velocity times force and so on. So for the large scale model, we could do it, but on the molecular level, it would be difficult.
Other questions? I had one coming back to Stanka, and I got disconnected for a second, so if I missed it, I mean, the speed of coming back to this question of optimization, which is the theme of your talk, the speed of PCR test reporting has improved a lot recently. And so I wondered how if you were going to, you talked about economic efficiency. And I wondered what the payoff for, say, decreasing the time for your PCR test reporting by an hour is. In other words, is that something that you really want to invest in? That if you could speed up your PCR tests? I know I had to have one done to come back to the United States a few weeks ago, and I got my results in 6 hours for PCR test.
Yeah, so
and I know in some places they're even faster. How significant is that, and how does that affect the?
right. So when we were doing the second study, in the one in which we're looking at vaccines, we used 1 day because almost everybody does it in 1 day. But you're right. If you're decreasing things, to 6 hours compared to an Abbott test of 15 minutes, you might do better with PCR almost always. Now you still need to go to a specialized doctor and at the time to get that thing done. But yeah, PCR might be your better option for 6 hours. But we haven't got to those fine-grained details on hours, right? We assume that it's 1 day because that's usually what people tell you. But yeah, I had one done not too long ago, and it was less than 12 hours. So yeah, we're gotten a lot better about it. But there's a lot of places in the world where they are still seeing really long delays.
So Stanza, you also mentioned at some point these LAMP tests, which the LAMP system, which is sort of in between, okay? Did you also look into that one as a viable alternative?
No, because almost all the standardized tests just look in the upper tract, right? So all of them are swabs, or all of them are nasal washes, or things like that. So you look into it. We also, once you get into the lung, it's not so much about the virus anymore. It's about whether or not you have way too much activity in terms of immune cells, and we're looking at that from the other point of view. Can I see that you're going to get bad outcomes, or are you going to get hypoxic at some point, or things like that, but not from the point of view of testing.
Okay.
Almost nobody's using that, for travel or anything like that. Yeah, they are looking at also at the beginning. They were looking at all sorts of things. So, for example, we're doing wastewater, right? So,
okay. Yeah.
right, people were doing things like looking in the stool, as well as the respiratory tracts. But you get fewer information from there, but you still get information. So, for example, if you're looking at wastewater on campus, we know whether or not a dorm has it. We might even, if we're really good about it, we might even know which floor of the dorm has the outbreak.
Yeah.
So yeah, but no, almost everything is swabbing. Nasal wash is painful, so even that is not an option. It's a nice alternative because you get a lot more virus, so you get fewer false negatives in that case. So.
I mean, we had a lot of the Salivary tests in Heidelberg. But the problem is that they're not officially approved, and in the end, you had to go for PCR anyway. Okay, so
right, yeah, and I mean, even for travel, right? So we're saying, well, if you have a PCR that's going to take a long time, take an antigen test, but not the one that you can administer yourself. You have to be supervised, so you might be able to administer it yourself, but over the phone with someone qualified on the other end. So we're not there yet in which, we can say we have all of these alternatives in which, as a responsible citizen, I can make up for the cost or time or things like that PCR comes with.
Okay, other questions? I think my Zoom session is locked up, so I can't see people. So if you want to say something, please speak up. I can talk in here, but I can't control the Zoom session at the moment. I mean, I think it was interesting when you were talking about pooled sampling. I guess pooled sampling doesn't work if the incidence level is very high. I mean, if every pooled sample is positive, you don't learn much. Right.
And it was something that everybody was considering at the beginning, where, we had tons of cases and you didn't have enough time or energy or people to test everybody. But yeah.
I mean, I'm wondering a little bit. I know now there's an oral approved antiviral. I now know 3 people who died from breakthrough cases after double vaccination. And I wonder if the kinds of studies you're doing might be able to do this question of very early how early can you do detection? Might be interesting, and also maybe something you showed, of course, viral load curves, or this sort of imputed viral load days after infection. If you include heterogeneity in that, I don't know if you did that in your model.
We did not. We assume everybody's the same.
Because that might have a big effect, because just the way you could have super spreader events, if one individual who's vaccinated has a much higher viral load, or at least distributable viral load, that could be significant in terms of it could dominate the kinetics of the model.
It's quite interesting, because if you're looking at it from a mathematical point of view, the slope of increase is really fast, right? So you get lots of viral load very early on. And when you're looking at the other end, it's a lot slower. So yeah, you can get someone that's infected in the first day spreading quite a bit. But now, when we're doing this, we're putting everything into the transmission rate. But I'm not sure if the transmission rate has to do with a person that's, I don't know, the soul of the party, right, in some super spreader event, or whether it has to do with, the actual virus doing better inside of an individual at that moment in time. So, all of that are lumped together in all of our statistics right now, and it would be very nice to be able to tease them out. And that's why I worry when people are making comparisons by comparing someone that got infected in 2021 versus someone that got infected at the end of the 2020, when all of us, made peace with the idea of masking, and now we're not anymore, right? Is it because I lowered the security that I'm creating a super spreader event, or is this virus inherently better,
Looking for questions. Again, I can't see the frame, so you have to speak up if you have a question. I have one question coming back to Ulrich. You mentioned there are 5 species of malaria parasite in humans. Have you done any comparative studies about whether the motility and speed are different in different species of malaria?
Oh, actually, the data I showed you today is only Plasmodium berghei, which is the mouse one. So I don't know. I mean, you see, if you work with human malaria, you have to go to safety level 3. And I mean, for the skin, I think nobody's doing that. Okay. In the blood stage, you are safe because you can propagate the parasite in blood as a culture. Biosafety level 1 is just fine. Okay. So there, everybody works with falciparum. But for the skin, you basically work in a mouse.
And you mentioned one strain, but there is only one strain that's used. I mean, presumably, yes. Is it known that there really is only one strain, or is it only that there's only one strain that's used in the lab?
Okay, I think there are more than one, but this is the one which everybody uses.
If we look at the other apicomplexa, are there other? I mean, you talked about toxoplasma.
Yeah.
Are there other apicomplexa that have the same kind of physical shape of the propagating parasite and that move in a similar way, or is that unique to malaria?
Okay, this I honestly don't know, but let me mention that the trypanosomes, which cause the sleeping sickness, they also move by basically flowing their surface. Okay, so they also have this sort of polar flow on the surface. So also they don't have this directional movement. They just want to move probably more to strip off the antibodies, but still it's interesting that there are slender structures with the surface flow. This seems to be a relatively general principle.
I mean, that's something, if we could engineer a device that moved with that flowing surface, it would be an amazing engineering feat, because you could have something that could move in any direction in any way.
But the so-called self-phoretic particles are a little bit like this, okay? So you have these metal particles which are coated with a little cap or something like this, okay? And then you have a surface flow from the front to the back. So it's not that different, okay? It's of course not lipids flowing, but some kind of chemical, but it's the same principle, I would think.
Right, at the sort of nanofluidic, microfluidic level, but we can't build an automobile that works that way.
Yes, I agree.
Yes, Janus particles. I mean, I remember Janus particle studies already when I was a graduate student, which is quite a while ago.
Yes.
And also you can do that electrophoretically. If you take a tiny chip, which is a diode, and you put it in a fluid with ions, and then you apply an alternating electric field, it'll crawl.
Yes.
So yes. No, I was thinking macro rather than micro.
Actually, and if you don't take a sphere, but something which is bent, it goes in a spiral. because you break the symmetry. And there's very nice work from the physics community on the shapes you get. But in the end, it's usually spirals. Okay, it's the general solution to this problem.
Lorenzo, did you have a final question?
Yeah,
We're out of time.
that,
Last
actually, out
question.
of all discussion, still to Ulrich, is might be very naive or might be actually been answered already. Might be just the shape of this structure that generate the spiral. So if otherwise, if they were spheres, would they do the spirals?
Yeah, no, it's not enough, because it's not only the spiral, it's also the chirality of the spiral. Okay, they only, I mean, on the glass, they only go counterclockwise. And this you can't explain by the shape itself. But there are several elements which might create a chirality. So I showed this ring, they have this, the apicomplex of the ring. The ring is not vertical, but it's tilted. Okay, and then the microtubule basket in Toxoplasma, it's very clear. It has a chirality, it's basically bent. Okay, in Malaria, in Plasmodium, it's not tilted, but there is also something. There are two microtubules, or is it one on one side, which are a little bit different than the other one on the other side. Okay, and this also creates some asymmetry. So there are several structural elements which might create this chirality, but not a shape per se.
I was asking that also because, well, this is a very, again, experimental observation done in a completely different system. If I do I culture endothelial cells normally.
Yeah.
So if I expose to certain factors, they become elongated. And when they become elongated, very often they also assume kind of a spiral shape. I always look at the images that are still, so I don't know about the dynamics of that. But giving a look around, it looks like there are kind of developing this kind of movement.
Interesting.
I was wondering if
yeah, so I
have
collected a few papers on this subject. It also catched my attention. And there are several phenomena in this direction which go back to the chirality of the actin. Okay, because actin is sort of a, if you wish, a helix, okay, like a double helix, and it has, of course, a chirality. And actin is usually polymerized out of some nucleator like formins, okay? And if the formin is nucleating on an actin, it's actually rotating. But if you attach the formin to some surface, then the actin is rotating, okay? And this is probably the normal situation. So normally the formins are attached to, let's say, a adhesion contact, and then the actin growing out of this is rotating, okay? And Sasha Vershatsky from the Mechanobiology Institute at Singapore, he has shown that this can be translated into a whole cell rotation if there's a clear-cut situation with, let's say, you have many adhesion contacts which are relatively well arranged to each other, then altogether it creates a rotation of the whole cell, okay? And even the nucleus sometimes rotates. And it really can be traced back to the actin. And there's an extremely interesting paper in Current Biology where they showed that if you do mutations on the formins, you can even turn the turn of a snail in the other direction. So even even something as macroscopic as the shell of a snail comes from the actin somehow, okay? And actually, a very similar observation is that in early development, okay, you have a rapid sequence of cell divisions, and there you often have to make decisions, is the next order left or right, okay? And this often is also determined by the actin system. So I think the chirality of the actin filament is at the base of many of these phenomena.
Thank you.
Yeah.
Okay.
But not in the plasmodium, because in the plasmodium, the actin is not so regularly arranged, okay? So it must be different. Okay. But for human cells, it's really interesting, or for snails or whatever.
Well, I want to thank then both of our speakers, especially for you staying up late for us. We appreciate it. Thank you for the diversity of talks. And I was delighted to have the discussion between our speakers today. I'm always glad to find common interests in the diversity. One of the things that I like so much about these mini seminars is just how varied the topics are related to infection and response. And so with that, I'd like to thank our speakers again for 2 great talks. And I'll call the meeting to a close for today. And I hope those of you who are here still, we'll see you next week. And of course, you're welcome to come back and talk more about your topics. We'd love to hear other projects on parasitic infection and on the other things that you're both doing. So if you'd like to speak again, let us know and we'll get you on the schedule. Okay,
Thanks
thank
a
you
lot.
so much. Okay,
Okay, have a good day. Bye-bye.
Bye.